Chapter 3: Biological Foundations of Intelligence. A comprehensive science of life must account for the nature as well as the variety of human intellectual competences. In view of the spectacular progress of recent decades in such areas as biochemistry, genetics, and neurophysiology, there is every reason to believe that the biological sciences will eventually be able to offer a cogent account of these intellectual phenomena. Indeed, it is high time that our understanding of human intellect be informed by the findings that have accrued in the biological sciences since the time of Franz Joseph Gall. And yet, because psychologists and biologists inhabit different environments, the task of marshaling biology to explain human intelligence has barely begun. As I read current findings in the brain and biological sciences, they bear with particular force on two issues that concern us here. The first issue involves the flexibility of human development. The principal tension here centers on the extent to which the intellectual potentials or capacities of an individual or a group can be altered by various interventions. From one point of view, development may be viewed as relatively locked in, preordained, alterable only in particulars. From an opposing perspective, there is far more malleability or plasticity in development, with appropriate interventions at crucial times yielding an organism with a far different range and depth of capacities and limitations. Also pertinent to the issue of flexibility are the related questions of the kinds of intervention that are most effective, their timing, and the role of critical periods during which pivotal alterations can be brought about. Only if such issues are resolved will it be possible to determine which educational interventions are most effective in allowing individuals to achieve their full intellectual potentials. The second issue is the identity or nature of the intellectual capacities that human beings can develop. From one point of view, which I associated earlier with the hedgehog, human beings possess extremely general powers, all-purpose information processing mechanisms that can be put to a large, or perhaps even an infinite number of uses. From an opposite perspective, that more reminiscent of the fox, human beings, like other species, have a proclivity to execute certain specifiable intellectual operations while proving incapable of performing other intellectual operations. An allied issue concerns the extent to which different portions of the nervous system are, in fact, committed to carrying out particular intellectual functions, as opposed to being available for a wide range of operations. It proves possible to analyze this issue of identity at various levels, ranging from the functions of specific cells at one extreme to the functions of each half of the brain on the other. Finally, as part of this concern with identity, the biologist needs to account for those capacities, like language, that seem to evolve to a high degree in all normal individuals, as against other capacities, like music, where striking differences in individual achievement are far more prevalent. Taken together, these sets of questions add up to a search for general principles that govern the nature and the development of human intellectual capacities, and that determine how these are organized, tapped, and transformed over a lifetime. Much current work in the biological sciences strikes me as relevant to these issues, though often it has not been conceived along these lines. Here I attempt to mine those lodes of research that seem most relevant to a student of the human mind. In my view, the preponderance of evidence points to the following conclusions. There is considerable plasticity and flexibility in human growth, especially during the early months of life. Still, even that plasticity is modulated by strong genetic constraints that operate from the beginning, and that guide development along some paths rather than along others. As for the issue of identity, evidence is accumulating that human beings are predisposed to carry out certain specific intellectual operations, whose nature can be inferred from careful observation and experimentation. Educational efforts must build upon a knowledge of these intellectual proclivities and their points of maximum flexibility and adaptability. These, then, are some conclusions at which one may arrive after weighing the relevant biological evidence. For individuals already familiar with the findings of the biological sciences, as well as for those who have little tolerance for reports from the harder sciences, it is possible at this point in the narrative to move directly to Chapter 4, where I introduce the criteria for an intelligence. Those with an interest in the details that undergird the foregoing conclusions are invited to enter the realm of genetics. Once one has elected to peer through the biologist's lens, an initial concern with genetics is virtually inevitable and certainly proper. Moreover, given the incredible strides made in genetics, since the code was cracked by James Watson and Francis Crick some 30 years ago, it is not surprising that psychologists have searched in the composition of DNA, RNA, and their fascinating interaction for the answer to puzzles about intellect. Unfortunately, however, lessons from this line of study are far from direct. To be sure, the geneticist's discoveries must form the point of departure for any biological study. After all, we are living organisms, and in one sense, everything that we will ever achieve has been coded in our genetic material. Moreover, the distinction between the genotype, the organism's makeup as determined by the genetic contributions of each parent, and the phenotype, the organism's observable characteristics as expressed within a given environment, is fundamental to consideration of any individual's behavioral and intellectual profile. Equally fundamental is the notion of variation. Because of the huge number of genes contributed by each parent and the innumerable ways in which they can be combined, we need not worry that any two individuals, except identical twins, will unduly resemble one another, or that any two individuals will exhibit identical profiles. Genetics has made its greatest progress in accounting for simple traits in simple organisms. We know a great deal about the genetic basis for structures and behaviors in the fruit fly, and through studies of inheritance patterns, we have gained insight into the transmission of specific human pathologies like sickle cell anemia, hemophilia, and color blindness. But when it comes to more complex human abilities, the capacities to solve equations, to appreciate or create music, to master languages, we are still woefully ignorant about the genetic component and its phenotypical expression. First of all, these abilities cannot be studied experimentally in the laboratory. Furthermore, rather than being related to a specific gene or a small set of genes, any complex trait reflects many genes, a fair number of which will be polymorphic, allowing a number of different realizations across a range of environments. Indeed, when it comes to capacities as broad and vague as human intelligences, it is questionable whether one ought to speak of traits at all. Those scholars of a geneticist bent have, of course, speculated about what a talent might be. According to one such account, certain combinations of genes may be correlated with one another, and these in turn may cause the production of enzymes that affect specific structures in one region of the brain. As a result of enzyme action, these structures might become larger, feature more connections, or promote more inhibition, any one of which possibilities might culminate in a greater potential for high achievement. But the very fact that these speculations involve so many hypothetical steps indicates how far they are from being established facts. We do not even know whether those individuals who have a talent, or for that matter a glaring defect, reflect an inherited tendency to form certain neural connections, which would then be found in others who are closely related, or whether they simply represent one tail of a randomly generated distribution, in which case they could equally well arise in any two unrelated individuals. Probably the most reliable clues to the genetics of human talents come from studies of twins. By comparing identical twins with fraternal twins, or identical twins reared apart with those reared together, we can gain some purchase on which traits are most subject to hereditary influences. Still, scientists eyeing the same set of data, and not even disputing the data, can reach widely divergent conclusions about heritability. Simply on the basis of certain mathematical and scientific assumptions, some scientists would place the heritability of intelligence, as measured by IQ tests, as high as 80%. In other words, these authorities would claim that up to 80% of the variability in intelligence scores in that population can be ascribed to one's genetic background. Other scientists pondering the same data but operating on different assumptions would estimate heritability as less than 20% or even zero. Naturally, most estimates run somewhere in between, with 30% to 50% being commonly cited. There is considerable agreement that physical traits are most straightforwardly genetic, that aspects of temperament are also largely genetic, but that when one comes to aspects of cognitive style or personality, the case for high heritability is far less convincing. The genetic literature provides few unequivocal answers to questions that preoccupy a student of intelligence. Nonetheless, there are useful concepts that might inform our investigation. Let's begin with the well-documented fact that, as a result of their genetic makeup, certain individuals are at risk for a given disease, like hemophilia, or neurological condition, like severe retardation. This fact does not in itself certify that they will get the disease. Factors of probability, as well as accidents of environment or special treatment, also play their role. This fact suggests only that, other things being equal, these individuals are more likely to contract the disease than is someone without this hereditary disposition. By analogy, it may be useful to consider certain individuals as at promise for the flowering of a certain talent. Again, this diagnosis does not ensure that they will develop the talent. One will not become a great chess player or even a patzer in the absence of a chessboard. But given an environment where chess is played and some stimulation, individuals of promise have a special proclivity for acquiring the skill rapidly and reaching a high level of competence. Being at promise is a sine qua non for becoming a prodigy. Even so, thanks to certain hothouse training methods, such as the Suzuki Violin Talent Education Program, even an individual with apparently modest genetic promise may make remarkable strides in a short time. Another suggestive line of speculation concerns the variety of traits and behavior of which human beings are capable. In a large and heterogeneous population like ours, with considerable intermarriage, one encounters a wide variety of traits, but over time, extreme traits tend to become invisible or to disappear altogether. In contrast, certain populations, for example those in isolated South Sea islands, have lived as a single group for thousands of years and have avoided any kind of intermingling with other groups. These latter populations will exhibit genetic drift. Through processes of natural selection, they will come to possess a genetic pool that may be distinctly different from that of other populations. It is not always possible to separate out the purely genetic factors from those reflecting an unusual natural environment or an exotic cultural system. Yet, according to the virologist Carleton Gajdusek, who has studied many primitive societies, populations subjected to genetic drift often exhibit a remarkable set of characteristics, including unusual diseases, immunities, physical traits, behavioral patterns, and customs. It becomes crucial to record these factors before they disappear or become invisible, owing to the demise of the group or to its extensive intermingling with other populations. Only by careful documentation of these groups will it become possible to determine the full range of human abilities. Indeed, once these groups have disappeared, we may not even be able to envision that they could have been capable of the actions, skills, or traits that they in fact exhibited. But once we have established that some group, in fact, that any human being, has exhibited a certain competence, we can look for and attempt to foster that capacity in other members of the species, our species. An opposite course can be pursued in the case of undesirable traits or competences. To be sure, we can never definitely establish that a given trait has a genetic component, but that determination actually proves irrelevant to our present concern. Documentation of the varieties of human nature. Consider this perspective in another way. Our genetic heritage is so variegated that one can postulate all kinds of abilities and skills, as well as maladies and infirmities that have not yet emerged, or that we have not yet come to know about. Given genetic engineering, countless other possibilities arise as well. An individual with a clever imagination might well be able to anticipate some of these possibilities. However, it is a far more prudent research strategy to sample widely among human beings of diverse stock and to determine which competences they have in fact achieved. Studies of remote and isolated groups, the prize for the geneticist, prove extremely valuable for psychologists as well. The broader the sampling of human beings, the more likely that any list of the range of human intelligences will be comprehensive and accurate. While genetics still proves to be of limited utility for the student of intelligence, a review of neurobiology, including the specialties of neuroanatomy, neurophysiology, and neuropsychology, promises to bear much richer fruit. Knowledge of the nervous system is accumulating as rapidly as knowledge of genetics, and the findings are much closer, so to speak, to phenomena of cognition and the mind. A study of neurobiology bears critically on the two central issues with which I am concerned in this chapter. We can ferret out general principles as well as specific nuances concerning the flexibility of development and the identity of human competences. In this survey, I shall begin by considering the issue of flexibility, and particularly those findings that document the relative plasticity of the nervous system during the early phases of development. Following the review of flexibility, I shall turn to those lines of research that help illuminate the abilities and operations of which human beings are capable. Though my concern throughout this volume will fall on the capacities of human beings and the extent to which they can be developed and educated through appropriate interventions, most of the findings that I review will, in fact, be drawn from research with animals, invertebrates as well as vertebrates. And in this effort, I have been helped in particular by two lines of work that have deepened our understanding of the principles of development. The work of David Hubel, Torsten Wiesel, and their co-workers on the development of the visual system in mammals, and the works of Fernando Notebaum, Peter Marler, Mark Konishi, and their co-workers on the development of singing capacities in birds. While the transfer from animal to human populations must be made cautiously, particularly in the intellectual realm, findings in these areas are far too suggestive to be ignored. A key concept for the understanding of neural growth and development is canalization. First posited by C. H. Waddington, a geneticist at Edinburgh University, canalization refers to the tendency of any organic system, like the nervous system, to follow certain developmental paths rather than others. Indeed, the nervous system grows in an exquisitely timed and elegantly programmed fashion. The origins of cells in the fledgling neural tube and their migration to regions where they will eventually constitute the brain and spinal cord can be observed with predictable regularity within and, to some extent, even across species. Far from representing a random or accidental collection, the neural connections that are actually affected reflect the highest degree of biochemical control. One beholds a stunning epigenetic sequence where each step in the process lays the groundwork for and facilitates the unfolding of the next. To be sure, the development of any system also reflects environmental influences. If, through experimental intervention, one alters the chemical balance, one can affect the migration of particular cells, or even cause one cell to carry out the function ordinarily assumed by another. Yet, according to Waddington, it proves surprisingly difficult to divert such patterns from what appears to be their prescribed developmental goals. In the present case, an adequately functioning nervous system. As Waddington put it is quite difficult to persuade the developing system not to finish up by producing its normal end result. Even if one seeks to block or otherwise to divert the expected patterns, the organism will tend to find a way to finish up in its normal status. If thwarted, it will not return to its point of origin, but will rather make its peace at a later point in the developmental course. So far, my description of the development of the nervous system has stressed strict, genetically programmed mechanisms. This is appropriate. And yet, an equally amazing facet of biological development is its flexibility, or to adopt the more technical epithet, its plasticity. An organism exhibits plasticity in a number of ways. To begin with, there are certain developmental periods where a relatively wide range of environments can each bring about the proper effects. For example, if a human infant is swaddled for most of the first year of life, it will still walk normally during the second year. Moreover, in the event that the young organism is deprived or damaged in a significant way, it may often exhibit great recuperative powers. Indeed, in general, this plasticity proves greater at the earliest points in development. For example, even if a human infant loses the dominant of its two cerebral hemispheres, it will still learn to speak. But there is a point at which the Rubicon is crossed and plasticity is permanently on the wane. The adolescent or the adult who loses a hemisphere will be severely impaired. Yet even these generalizations about plasticity must be qualified. First of all, sometimes early injury or deprivation can have extremely severe results. E.g., failure to use one eye during the first months of life infirms the possibility of binocular vision. Second, certain abilities or skills prove robust, even in the face of an adult injury, thus suggesting a residual plasticity that endures throughout much of the life cycle. Some adults recover the ability to talk despite massive injury to the left or dominant hemisphere of the brain, and many recover the use of paralyzed limbs. Overall, the notion of plasticity cannot be considered apart from the timing of a particular manipulation or intervention and from the nature of the behavioral competence involved. Plasticity is limited in other ways as well, reflecting their behaviorist heritage. Some psychologists have been prone to assume that most any organism can, given proper training, learn to do most anything. Searches for horizontal laws of learning have often reflected this faith. Similar claims have been made about human beings, such as the suggestion that anything can be learned at any age in some useful form. More recent studies have come down, however, in harsh opposition to this optimistic cast of mind. An emerging consensus insists that each species, including ours, is specially prepared to acquire certain kinds of information, even as it proves extremely difficult, if not impossible, for that organism to master other kinds of information. A few examples of this preparedness and counter-preparedness will be helpful. We know that many birds are capable of learning songs, and some can eventually produce a great variety of songs. And yet female sparrows may be so carefully pre-tuned that they are sensitive only to the particular dialect sung by males in their own region. Rats can learn very quickly to run or jump to escape shock, but only with greatest difficulty to press a lever in order to effect the same escape. Moreover, there are even limitations to the jumping mechanism. While jumping to avoid a shock seems to be a natural or prepared response, if the rat must jump in a box with a closed lid, learning will be extremely slow. The ease with which all normal and many subnormal individuals master natural language, despite its apparent complexity, suggests that the species as a whole is specially prepared to acquire facility in this realm. By the same token, the difficulties most humans exhibit in learning to reason logically, particularly when propositions are presented in an abstract form, suggests no special preparedness in this area and perhaps even a predisposition to attend to the concrete specifics of a situation, rather than to its purely logical implications. While no one understands the reasons for selective preparedness, it may be that certain neural centers can be readily triggered, stimulated, programmed, and/or inhibited, while others prove far more difficult to activate or inhibit. In the light of these general remarks about plasticity and behavior, we are now in a position to look more closely at evidence pertaining to the degree of determinedness or flexibility that characterizes the developing organism. Our survey will consider the particulars of plasticity around the time of birth, the effects of various early experiences on subsequent development, and the possibility that various kinds of learning can be understood at the neurological or the biochemical level. While the particulars concerning each species differ, research on plasticity in early life has yielded a number of principles that seem reasonably robust. A first principle enunciates the maximum flexibility encountered early in life. Consider one example which can stand for many others in the literature. As explained by W. Maxwell Cowan, neurobiologist at the Salk Institute, both the forebrain and the neural part of the eye develop out of the head end of the neural plate. If, at an early stage in development, one removes a small piece of ectodermal tissue, neighboring cells proliferate, and the development of both the brain and the eye should proceed normally. But if the same operation is performed a little later, there is a permanent defect either in the forebrain or in the eye. The actual damage depends on the particular piece of tissue that has been removed. Such progressive blocking out leads to the determination of increasingly precise regions of the brain. Studies by other neurobiologists, such as Patricia Goldman, confirm that during the earliest period of life, the nervous system can adapt flexibly to severe injury or experimental alteration. For some time thereafter, the nervous system may devise an alternative route or connection that may prove adequate. But if the injury or alteration occurs too late during the developmental process, the relevant cells will either connect randomly or atrophy altogether. A second related principle underscores the importance of so-called critical periods during the process of development. For example, in the cat, there is a critical period in visual development from the 3rd to the 5th postnatal week. If, during this time, one eye is deprived of form or light, then the central connections of the eye change and the ill-seeing eye will be suppressed from functioning. Such interference seems to be permanent. As a general point, it seems that the most vulnerable time for an organism occurs during these sensitive periods. Irreversible damage to the central nervous system seems particularly likely to occur in the wake of even mild restrictions during such a critical period. Conversely, rapid development will occur if the proper conditions are obtained during the critical period. According to a third principle, the degree of flexibility differs across the region of the nervous system with which one is concerned. Regions that develop later in childhood, such as the frontal lobes or the corpus callosum, turn out to be more malleable than those that have developed in the first days and weeks of life, such as a primary sensory cortex. The surprising degree of uncommittedness that characterizes regions like the corpus callosum seems to reflect both the need for a high degree of modifiability for certain cortical connections and the importance of specific postnatal experiences in determining the kinds of connection that will ultimately be made. Indeed, when it comes to the most complex of human capacities, such as language, the individual can withstand even massive damage, including the removal of an entire hemisphere, during the first few years of life and still acquire the ability to speak in a reasonably normal fashion. This recovery suggests that large portions of the cortex remain uncommitted and thus available for diverse uses during early childhood. A fourth principle concerns those factors that mediate or modulate development. An organism will fail to develop normally unless it undergoes certain experiences. Thus, the cat's visual system will not develop normally, and parts of it will actually atrophy if the animal is not exposed to patterned light after birth. Moreover, the cat must be exposed to a variegated environment, permitted to use both eyes and move about its environment. If exposed to horizontal patterns only, the cells destined to carry out vertical processing will either atrophy or be taken over for other functions. If the cat is allowed to use only one eye, those cells dedicated to binocular vision will degenerate. And if the cat does not move actively in its environment, for example, if it is passively transported through a patterned environment, it will also fail to develop a normal visual system. It seems that the neural system that mediates vision exhibits a scheduled plan for development that expects visual inputs of certain sorts during sensitive periods. If proper stimulation is lacking, or if inappropriate stimulation is supplied, the usual developmental goals will not be achieved, and the animal will fail to function properly in its environment. A final principle treats the long-term effects of injury to the nervous system. While some injuries exert immediate and evident effects, others may be invisible at first. Suppose, for example, that a region of the brain that is destined later in development to assume an important function happens to be injured at an early point in life. It may well be that the consequences of the injury will not be observed for some time. Thus, injuries to the frontal lobes in primates may not be detected during the first years of life, but may become all too manifest at a later time when the animal is expected to carry out those complex and organized forms of behavior that are ordinarily mediated by the frontal lobes. Early brain injury may also stimulate certain anatomical reorganizations that ultimately prove counterproductive. For instance, connections may be formed that allow the animal to carry out tasks crucial at the present time, but that prove useless for the emergence of needed skills at a later time. In such cases, the tendency of the nervous system to canalize may actually have maladaptive long-term consequences. Consideration of these 5 principles should confirm that any simple verdict on the issue of determination versus flexibility is impossible. Strong pressures favor each factor, and both therefore exert considerable influence on the development of the young organism. Determination, or canalization, helps to ensure that most organisms will be able to carry out the functions of the species in the normal way. Flexibility, or plasticity, allows for adaptability to changing circumstances, including anomalous environments or early injuries. Clearly, if one must sustain an injury, it is better to have it early, but probably every deviation from the normal developmental path exacts its cost. Up to this point, I have considered chiefly the effects of specific kinds of experience, such as exposure to certain kinds of stripes, upon relatively circumscribed regions of the brain, most notably the visual system. But psychologists and neurologists have also examined the question of more general effects of stimulation, or deprivation, on the overall functioning of organisms. Pioneering studies come from Mark Rosenzweig and his colleagues at the University of California at Berkeley. Beginning in the early 1960s, the Rosenzweig group raised animals, chiefly rats, in environments that were enriched. These environments featured numerous other rats, as well as various toys such as ladders and wheels. A comparable set of rats were raised in an impoverished environment, which contained enough food but no special features. The enriched rats performed better on various behavioral tasks and were also trimmer than their well-fed but duller peers. After 80 days in these contrasting environments, all the rats were sacrificed and their brains subjected to analysis. The crucial findings: the cerebral cortexes of the enriched rats weighed 4% more than the cortexes of the impoverished, though fatter, rats. More crucially, the largest increase in brain weight occurred in those portions of the brain that serve visual perception, presumably the portions that had been particularly stimulated in the enriched environment. Numerous studies with rats and other species have confirmed that an enriched environment produces more elaborate behavior as well as palpable changes in brain size. Effects can be surprisingly specific. The Rosenzweig team has shown that if you provide a richer experience to only one half of the brain, only that half exhibits changes in cell structure. William Greenough has demonstrated that in animals raised in complex environments, one finds larger nerve cells in some brain areas, as well as more synapses, synaptic connections, and other dendritic connections. As he summarizes it, the gross regional size changes that accompany differences in experience are associated with changes at the neuronal level in the number, pattern, and qualities of synaptic connections. Other intriguing and highly specific brain changes have been claimed. As part of his studies of the songs of male canaries, Fernando Nottebohm has correlated the size of two nuclei in the bird's brain with the appearance of singing. He finds that during the most productive vocal periods, these two nuclei may become double the size they reach during the least productive period, during the summer molt. Then, when the brain grows larger in the fall, new nerve fibers develop, fresh synapses are formed, and accordingly, a larger song repertoire once again emerges. Apparently, in birds, the learning, or relearning, of a motor activity translates directly into the size of the relevant nuclei, the number of neurons, and the extent of connections among them. To my knowledge, scientists have hesitated to speculate in print about similar changes in brain size accompanying or causing the diverse ability profiles in human beings. In the absence of suitable experimental methods, such prudence seems proper. But it is worth citing the observation of two talented neuroanatomists, O and A. Vogt. For many years, the Vogts conducted neuroanatomical studies of the brains of many individuals, including talented artists. One painter whose brain they observed turned out to have a very large fourth layer of cells in his visual cortex, and a musician with perfect pitch from early childhood had an analogously large region of cells in his auditory cortex. As hypotheses of this sort become more respectable, and as non-invasive methods for studying brain size, shape, and processing routes become more widely utilized, I would not be surprised to find contemporary support for these ancient phrenological themes. But bigger is not always better, and vast numbers of cells or fibers are not always virtues in themselves. Indeed, one of the most fascinating findings of recent years in neurobiology supports this cautionary note. Initially, the nervous system produces a great excess of neuronal fibers. A significant portion of the developmental process involves the pruning or atrophying of excessive connections that do not appear to be necessary and may, in fact, be injurious to normal function. The most critical interpretations along these lines have been made by two French scientists, Jean-Pierre Changeux and Antoine Donchain. These researchers have noted that in diverse regions of the brain, there are many more neurons initially than will eventually survive. A period of selective cell death occurs, usually at about the time when the population of neurons are forming synaptic connections with their designated targets. The death may involve anywhere from 15% to 85% of the initial neuronal population. Why should there be a large excess of initial connections? And why should certain connections endure while others atrophy? There is speculation that early excessive sprouting reflects, or better constitutes, the flexibility of the period of growth. This normal feature of development also has adaptive advantages. If some damage occurs during a time when excessive connections are available, the chances are greater that the organism will survive despite injury. In support of this notion, a tremendous growth in cell connections occurs immediately after a lesion, with sometimes as much as 6 weeks worth of growth occurring in 72 hours. Analogously, if one eye is removed at birth, the death of retinal ganglion cells, which would ordinarily occur in the first 2 postnatal weeks, is markedly reduced. There are other possible reasons for this proliferation of cell processes and synapses. During this period of richness, there appears to be intense competition among the cells, with those that are most effective in forming connections of the appropriate strength and specificity proving most likely to survive. Perhaps in time-honored Darwinian fashion, this proliferation may entail a kind of competition, allowing the most fit or appropriate of the organism's nerve fibers to prevail. An excess of nerve fibers in early development may well lead to the transient appearance of functional and behavioral properties associated with the excess connections. And here one may encounter U-shaped phenomena where the behavior of the immature organism, the left arm of the U, bears a striking resemblance to behavior ordinarily found only among adult organisms, the right arm of the U. Quite possibly, certain early reflexes in humans, like swimming or walking, reflect a proliferation of connections that temporarily allow certain precocious behavior. It is also possible that the tremendously rapid learning of which young organisms are capable, particularly during certain critical periods, may be related to an excess of connections, some of which will soon be pruned or eliminated. For instance, in human beings, the density of synapses increases sharply during the first months of life, reaches a maximum at the ages of 1 to 2, roughly 50% above the adult mean density, declines between the ages of 2 and 16, and then remains relatively constant until the age of 72. More than one scientist has speculated that the extremely rapid learning of the young child, for example in the area of language, may reflect an exploitation of the large number of synapses available at that time. And after the pruning has occurred, we may have here a functional definition of maturity, that time when excess cells have been eliminated and the originally targeted connections have been affected. The flexibility of youth seems at an end. Through survival of the fittest, the number of neurons has now been adjusted to match the size of the field that they are designed to innervate. If a new target like an extra limb is surgically added, the number of neurons will not decline as precipitously. There is now additional space in which the synapses can be formed. The critical period apparently ends when the process of synapse elimination has progressed to the point where few, if any, synapses are still capable of competitive interaction. Most scientists feel that there are further neural changes later in life. But whether with aging there is a gradual decline in synaptic density, progressive reduction in dendritic length and branching, or a more selective loss being restricted to certain areas of the cortex, is an issue on which scientists are not yet agreed. Understandably, most neurobiological research with implications for human beings has been conducted with primates, and there has been a focus on the principal sensory systems, which presumably work similarly across the biological order. But recently there have been efforts to understand the basis of learning, using species quite remote from man. Because of the suggestiveness of the findings, I quote 2 samples here. In my view, a veritable treasure trove of information that has stimulated the thinking of cognitively oriented scholars has been obtained by students of birdsong. Although from a different order of animals, birdsong involves highly complex activities, which, as it happens, are lateralized to the left portion of the bird's brain and are all mastered in instructive ways during the juvenile period. In the face of many differences across bird species, a few generalizations seem warranted. Early in the first year of life, the male bird produces a subsong, a babbling output that continues for several weeks. This is followed by the period of plastic song, a longer interval, where the bird rehearses a large number of the bits of those songs that it will eventually use to communicate its territory to other birds and also to advertise for a mate. This playful rehearsal resembles the exploratory activities exhibited by primates in many realms of activity. Where avian species differ from one another is in the flexibility and the conditions of song learning. Some birds, like the ringdove, will come to sing the song of their species eventually, even in the absence of exposure to the correct model. Some birds, like canaries, require feedback from their own singing. but not exposure to a model. If such birds are deafened, they fail to master their species' repertoire. And other birds, like the chaffinch, require exposure to the models supplied by conspecifics in order to sing an appropriate song. Some birds sing the same songs each year, while others alter their repertoire on an annual basis. It is clearly vital to understand the biological underpinnings of these cultural differences. But what is striking is that the bird emits during learning far more songs and song bits than it will vocalize during its adult prime. Moreover, birds are oriented to favor the songs from the environment that their species is destined to learn, and, relatively speaking, to ignore songs from other species or even other dialects of their own species. As I have already noted, the production of song depends upon structures in the left portion of the bird's nervous system. Lesions there prove far more disruptive than lesions in matching areas of the right brain. One can, in fact, produce an aphasia or amusia in birds. But the aphasic canary can recover its prior songs because the homologous pathways of the right hemisphere have the potential of being exploited. In this recovery of function, birds are more fortunate than adult humans. The learning of bird songs provides one intriguing model for how organisms come to master a highly particular kind of skill through the interplay of environmental stimulation, exploratory practice, and a predisposition to develop certain structures of the nervous system. In my view, it may well prove possible someday to apply some of the principles involved in the learning of birdsong to the processes whereby higher organisms, including human beings, come to master the cognitive and symbolic systems of their own cultural milieu. Another quite divergent set of researches using a simple mollusk called Aplysia californica promises to cast additional light on the biological basis of learning. With his colleagues, Eric Kandel of Columbia University has been examining the simplest forms of learning in Aplysia. He has been investigating how this organism, with a relatively small number of neurons, becomes able to habituate to a stimulus, so that it no longer responds after a while, becomes sensitized to a stimulus, so that it can respond even in the presence of diminished stimulation, and becomes classically conditioned, so that it can respond to a learned or conditioned stimulus as well as to an unconditioned or reflexive stimulus. And he has come up with 4 major principles. First of all, elementary aspects of learning are not diffusely distributed in the brain, but can rather be localized in the activity of specific nerve cells. Some learned behavior may involve as few as 50 neurons. Second, learning results from an alteration in the synaptic connections between cells. Rather than necessarily entailing new synaptic connections, learning and memory customarily result from alteration in the strength of already existing contacts. Third, prolonged and profound changes in synaptic strength can come about through an alteration in the amount of chemical transmitter released at the terminals of neurons, the sites at which cells communicate with other cells. And so, for example, in the course of habituation, each action potential produces progressively less influx of calcium and, hence, less transmitter release than does the preceding action potential. Finally, these simple processes of altering synaptic strengths can be combined to explain how progressively more complex mental processes take place, and thus yield, in Kandel's phrase, a cellular grammar underlying various forms of learning. That is, the same processes that explain the simplest form of habituation serve as a kind of alphabet from which one can compose far more complex forms of learning, such as classical conditioning. As Kandel summarizes his findings, basic forms of learning, habituation, sensitization, and classical conditioning select among a large repertory of pre-existing connections and alter the strength of a subset of this repertory. An implication of this view is that the potentialities for many behaviors of which an organism is capable are built into the basic scaffolding of the brain and are to that extent under genetic and developmental control. Environmental factors in learning bring out these latent capabilities by altering the effectiveness of the pre-existing pathways, thereby leading to the expression of new patterns of behavior. Thanks to Kandel and the lowly mollusk, some of the major forms of learning investigated by psychologists can be described in terms of events occurring at the cellular level, even including certain candidate chemical transactions, The once seemingly unbridgeable gap between behavior and biology seems to have been sealed. The work of Condell and his associates also casts tantalizing light on those issues of the specifiability of particular competences, which will concern us in the following pages. It seems that the same principles may characterize all neural cells, independent of species membership or form of learning. Apparent support for a horizontal view of learning. And yet, as Condell indicates, organisms themselves seem capable of only certain patterns of behavior and not others. And this phenomenon will also have to be accounted for in any biological approach to cognition. In contrast to the state of affairs in genetics, where links between the mainstream of the science and issues of human cognition were few and speculative, the view from neurobiology has proved far more revealing for our enterprise. Clear principles of plasticity and flexibility, determination and canalization have been uncovered. There is good reason to believe that, with suitable modifications, these principles can apply to the ways in which human beings develop certain cognitive systems and learn to achieve certain intellectual skills in the process of following certain paths rather than others. The clear and manifest effects of rich, or impoverished, early experiences on the general functioning of the organism have also been convincingly demonstrated. And we already know from studies of the effects of malnutrition in human beings that analogous effects can occur in our own species, with deleterious consequences for both emotional and cognitive functioning. Finally, through studies of such unlikely populations as songbirds and California mollusks, we have received promising insights into the ways in which forms of learning are manifest at the nervous system, cellular, and biochemical levels. While there is still an enormous distance between these simple forms of behavior and the varieties of learning and development of greatest moment to human beings, at least some of the insights gained from these lines of study should eventually prove applicable to the investigation of human learning and mastery. Up to this point, I have at times tolerated a convenient fiction, the contention that the nervous system is relatively undifferentiated and that variation in size, density, and connectivity can be discussed with seeming indifference to where these differences happen to be found. In fact, however, study of the nervous system has revealed an astonishingly highly organized architecture, with incredible specificity in appearance and in organization. Moreover, differences in organization appear to be closely linked to differences in the functions subserved by different portions of the brain. For example, it is clear that the earlier maturing areas of the cortex are involved in primary sensory functions, the perception of discrete sights and sounds, while the later maturing association sensory cortexes mediate the meanings of the stimuli and affect connections between sensory modalities. For example, associating seen objects with heard names. For our purposes, the organizational structure of the nervous system can be considered at two separate levels of detail: a fine-grained or molecular structure, and a grosser or molar structure. While this is also a convenient fiction, it is not a frivolous one. It was in fact recognized in the division of the 1981 Nobel Prize between Roger Sperry, in recent years a student of the molar level, and David Hubel and Torsten Wiesel, students of the molecular structure. And it proves highly relevant to our search for the identity of human intellectual functions. According to Vernon Mountcastle, physiologist at Johns Hopkins University, the human cerebral cortex can be viewed as being organized into columns or modules. These columns, which are vertical to the surface of the cortex, are approximately 3 millimeters long and between 0.5 and 1 millimeter across. They are increasingly recognized as forming separate anatomical entities that give rise to different quasi-independent functions. In fact, perception and memory may be distributed through the nervous system in the person of these special-purpose cognitive demons. These columns were first established in the visual cortex. As Hubel and Wiesel put it, given what has been learned about the primary visual cortex, it is clear that one can consider an elementary piece of cortex to be a block about 1 millimeter square and 2 millimeters deep. To know the organization of this chunk of tissue is to know the organization for all of the visual cortex. The whole must be mainly an integrated version of this elementary unit. In light of more recent findings, it seems probable that other sensory areas also consist of such columns, and it has even been proposed that the frontal lobe, the area deemed responsible for more abstract and less topographically mapped knowledge, has a columnar organization of this sort. To what sorts of things do these columns, or their constituent cells, respond? In the visual system, they respond to orientation, horizontal, vertical, oblique, and to ocular dominance, different degrees of eye preference. Less thoroughly understood cortical cells in the visual system may also respond to color, direction of movement, and depth. In the somatosensory system, the columns respond to the side of the field that has been stimulated and to the location of receptors in the skin layers. In the frontal lobe, the columns respond to spatial and temporal information concerning objects that have been present in the organism's field. Taken together, sensory and motor areas appear to contain two-dimensional maps of the world they represent. As information on vision or touch or sound is relayed from one cortical area to the next, the map becomes progressively more blurred and the information carried becomes more abstract. The columns may turn out to be the fundamental unit of organization throughout evolution. Columns have similar size and shape, not only within but also across species. Thus, different species of monkeys may have cortexes of different sizes and different numbers of columns, but the actual dimensions of their columns are the same. Patricia Goldman and Martha Constantine Payton have speculated that when the number of axons directed in a certain way exceeds a critical number, columns will form as a time-tested and efficient way of filling in the space. Indeed, if one plants an extra eye into a frog during gestation, a column will promptly be set up. But is the column or module the only way to think of the nervous system? Vernon Mountcastle, whose work led to the discovery of the columnar organization of the nervous system, himself distinguishes between mini-columns, which may have as few as 100 cells and thereby constitute the irreducibly small processing unit of the neocortex, and the macrocolumn, each packing several hundred mini-columns inside. Proceeding to a grosser level of organization, Francis Crick suggests the existence of larger distinct areas. The owl monkey, for example, has at least 8 cortical areas that are primarily visual. All are perceptually distinct, with a fairly well-defined boundary. In his opinion, there are perhaps 50 to 100 discrete areas in the human cortex. As he puts it somewhat wistfully, if each area could be clearly stained post-mortem so that we could see exactly how many there are, how big each one is, and exactly how it is connected to other areas, we would have made a big step forward. It seems clear at this point that one may divide the nervous system into units of widely different sizes. What is important for our inquiry is that, through the appearance and location of different neural units, nature provides important clues about the identity of its valued processes and functions. In speculating about larger areas of the cerebral cortex, we move to what has been called a molar level of brain analysis, a level dealing with regions that can be readily inspected by the naked eye. While molecular studies rely heavily on recordings from single cells, visible only under high powers of magnification, the chief source of information about molarity of mind comes from clinical work with brain-damaged patients. As a result of stroke, trauma, or other accidents, individuals may suffer damage to extensive but still limited regions of the brain. The frontal lobe of the brain may be destroyed wholly or partially, unilaterally. There may be a lesion in the temporal lobe or at the temporoparietal juncture. The results of this injury can be seen through radiological measures, brain or CAT scans, and can, of course, be examined with great precision in a post-mortem. Here is where the scientific payoff inheres. It becomes possible to correlate the loss of sizable portions of brain tissue, sometimes ones that are sharply delimited, sometimes ones dispersed across several regions of the brain, with specific patterns of behavioral and cognitive breakdown. By far the most excitement has been generated by the discovery that the two halves of the brain do not subserve the same functions. While each hemisphere controls motor and sensory capacities on the opposite side of the body, one side of the brain is clearly dominant. Such dominance determines whether an individual is right-handed, in the case of left brain dominance, or left-handed, in the case of right brain dominance. More telling for our purposes than this relatively mechanical division of labor, it has now been established beyond reasonable doubt that the left hemisphere is dominant for language in most normal right-handed individuals, while the right hemisphere is dominant, though not to the same extent for visual spatial functions. This much is known to all followers of biological science. What is less widely appreciated is that specificity of cognitive function can be tied much more precisely to finer regions of the human cerebral cortex. This state of affairs has been best worked out in the case of language. We find that damage to the frontal lobe, particularly that area called Broca's, results in selective difficulty in producing grammatical speech against a background of relatively preserved comprehension. Conversely, damage to the temporal lobe in the area called Wernicke's allows relatively fluent speech, replete with appropriate grammatical inflections, in the face of distinct difficulty in comprehension of language. Other, even more specific linguistic disorders turn out to be linked to particular regions in the brain. These include selective difficulties in repetition, naming, reading, and writing. It seems clear, then, that in the normal adult, cognitive and intellective functions of distinctiveness can be linked to particular areas of the brain that, in many cases, are morphologically distinct. David Hubel provides impressive testimony in favor of this point of view. We are led to expect each region of the central nervous system has its own special problems that require different solutions. In vision, we are concerned with contours and direction and depth. With the auditory system, on the other hand, we can anticipate a galaxy of problems relating to temporal interactions of sounds of different frequencies. And it is difficult to imagine the same neural apparatus dealing with all of these phenomena. For the major aspects of the brain's operation, no master solution is likely. Many of the issues raised earlier reemerge in the context of human cognition. For example, the left hemisphere is committed to being the language hemisphere, and yet, if that hemisphere must be removed early in life, most individuals will still acquire relatively normal language. To be sure, there are impairments in the language of a hemispherectomized patient, but it takes fairly subtle testing to reveal them. In other words, plasticity is evident in the acquisition of language, but such flexibility diminishes rapidly after puberty. Even a relatively punctate lesion in the left hemisphere of an adult male over the age of 40 is likely to produce a permanent and debilitating aphasia. The language zones may have been designed over time to serve auditory oral language, and yet they do not fall into disuse if an individual is deaf. Indeed, the zones that ordinarily subserve language can be mobilized in instructively different ways to acquire sign languages or other ersatz communication systems. For instance, deaf children who do not reside in signing households will on their own, or working in tandem, invent gestural languages. Careful analyses by linguists reveal clear-cut analogies between such contrived gestural language and the natural language of hearing children. For example, in the manner of construction of two-word utterances. Thus, there seems to be canalization not only in the exploitation of certain brain regions, but also in the functional lines along which a communicative system develops. Finally, if through severe abuse an individual should be deprived of language until puberty and then given an opportunity to learn to speak, there may be progress in some aspects of language. Yet, in the one case that has been carefully studied, this advance in language apparently occurred through exploitation of right hemisphere regions. Quite possibly, some sort of critical period for the mobilization of left hemisphere structures has elapsed, and so the language of this individual turns out to be most deficient in such left hemisphere aspects as grammar. While the story about other higher cognitive functions is not nearly so well understood, nor so clear, as that which obtains for language, there are determinable cortical organizations for other higher mental functions. And these functions, too, can break down along predictable lines. Adducing evidence for these other human intelligences is a task for the succeeding chapters of this book. At this point, however, it becomes germane to review some contrasting opinions about the relationship between intellect and brain organization, as well as certain efforts to relate human cognition to the specific computational capacities represented by the neural columns or regions. Discussion of the relationship between intellect and the brain has reflected prevailing scientific views about the general organization of the brain. At times when a localizationist view has held sway, there has been a correlative belief that different parts of the brain subserve different cognitive functions. Sometimes the discussion has focused on horizontal faculties. Perception is seen as residing in one region, memory in another, though more frequently the discussion has centered on specific vertical contents. Visual processing in the occipital lobe, language in the left temporal and frontal regions. During other scientific eras, the brain has been regarded as a general information processing mechanism and as an equipotential organ, where functions can be performed and skills represented in any section of the nervous system. At such times, intelligence has tended to be regarded, in hedgehog fashion, as a unitary capacity, one linked to the overall mass of usable brain tissue. 50 years ago, it appeared as if the holists or equipotentialists would carry the day. The influential work of the neuropsychologist Carl Lashley suggested that the amount of intact brain tissue, rather than its particular identity in terms of fissures and commissures, would determine whether an organism, like a rat, could perform a task. Lashley showed that one could section regions from almost any area of the brain, and the rat would still be able to run a maze. While this finding seemed to sound a death knell to localizationist positions, subsequent examination of this research has revealed a basic flaw. As the rat was relying on highly redundant cues from several sensory areas in running the maze, it really did not make much difference which areas of brain were removed, as long as at least some were left. Once investigators began to attend to the specific kinds of cues that were used, and to eliminate redundancies, even quite focal lesions turned out to impair performance. Parallel trends can be observed in studies of human cognitive processes as well. The bully period for the holists or generalists like Henry Head and Kurt Goldstein was half a century ago. Again, intelligence was related to the amount of brain tissue that was spared rather than to its particular locus. It does indeed seem that many human intellectual tasks can be carried out even in the wake of considerable brain damage. But again, once a task has been examined more carefully, its dependence upon certain key brain areas will ordinarily be demonstrated. Recent studies have revealed that the posterior parietal regions, particularly in the left hemisphere, prove of special importance in the solution of tasks thought to measure raw intelligence, such as Raven's progressive matrices. Even in the miasma of intelligence testing, it seems that the nervous system is far from equipotential. We find then an emerging consensus about brain localization. The brain can be divided into specific regions, with each emerging as relatively more important for certain tasks, relatively less important for others. Not all or none by any means, but with definite gradients of importance. In the same vein, few tasks depend entirely on one region of the brain. Instead, once one examines any reasonably complex task, one discovers inputs from a number of cerebral regions, each making a characteristic contribution. For example, in the case of freehand drawing, certain left hemisphere structures prove crucial for the providing of details, while right hemisphere structures are equally necessary for the mastery of the overall contour of the depicted object. Compromise in either half of the brain will result in some impairment, but the kind of impairment can only be anticipated once one knows where the brain injury has occurred. While this description of brain organization would be regarded as reasonable by most workers in neuropsychology, it has yet to exert much impact in cognitive psychology in this country. Neuropsychology is more widely accepted by cognitivists in Europe, possibly owing to the greater involvement there of medically trained scientific investigators. Indeed, as we have seen before, most workers in the cognitive area do not yet consider evidence about the organization of the brain relevant to their concern, or feel that neural processes must be made consistent with cognitive accounts rather than vice versa. To use the jargon, hardware is considered irrelevant to software, wet preparations to dry behavior. One encounters in American cognitive circles the continued belief in the existence of extremely general problem-solving capacities, which cut across every manner of content, and which presumably can be subserved by any area of the nervous system. Now, even if localization proves to be the most accurate description of the nervous system, it remains possible that there may still be very general problem-solving devices, as well as considerable horizontal structure, with perception, memory, learning, and the like cutting across heterogeneous contents. Nonetheless, it would seem high time for psychologists to take seriously the possibility that the molar, and even the molecular, analyses of the nervous system may have definite implications for cognitive processes. Approaching cognition from widely divergent perspectives, the philosopher Jerry Fodor, the physiological psychologist Paul Rozin, the neuropsychologist Michael Gazzaniga, and the cognitive psychologist Alan Allport have all endorsed the notion that human cognition consists of a number of special-purpose cognitive devices, presumably dependent upon neural hardwiring. There are many differences in emphasis in their accounts, some of which will be reviewed in Chapter 11, but for present purposes, the points of consensus can be expressed as follows. Over the course of evolution, human beings have come to possess a number of special-purpose information processing devices, often termed computational mechanisms. Some we share with some animals, face perception, while others prove peculiar to human beings, syntactic parsing.